Hi, I'm Sarah MacKell. I'm the lead biologist for Wildlife Preservation Canada's Native Pollinator Initiative. I'm here obviously with Mathilde Tissier, who is a Libero Aero postdoc with Laval and Bishop's University. And today we'll be presenting on our joint project, which is called Assessing Pollen Nutrition Impacts on Bumblebee Health and Reproduction. I would just like to start with a land acknowledgement. Since this program is being held virtually, a singular land acknowledgement does not capture all of our locations, obviously, and nativeland.ca can help you learn more about the land on which you find yourself. Carolyn also mentioned this website, but I just wanted to mention it again, just in case anyone missed it this morning. I will share the acknowledgement for the land surrounding African Lion Safari, since this is where Wildlife Preservation Canada's Bumblebee Conservation Lab is located and where most of the work discussed was conducted. Hamilton, Ontario is situated on the homelands of the neutral Haudenosaunee and Anishinaabe peoples. However, the WPC team works across Turtle Island, and we recognize the diversity of Indigenous peoples who have and continue to steward these lands.
Sorry, so and now it's my turn. And so as you all know already, humans are changing the composition, the diversity and the functionality of most ecosystems, and thus the diversity and the quality of food for wild animals that are often constrained to monotonous diets. This can have a strong effect on their performance by either affecting their internal state or their resistance and resilience to external threats such as pathogen exposure or climate change. And this will ultimately affect population dynamics. And animals' nutritional balance is at the core of these relationships and does not solely include intakes in proteins, fat, and carbohydrates, but also intakes of micronutrients such as vitamins and amino acids. And today we will focus on the vitamin B3 and its precursor, the tryptophan. And why is that? Well, for many reasons. First, because tryptophan and vitamin B3 are essential in all animals, from humans to invertebrates, and they must be obtained from the diet on a daily basis. They modulate key functions, key behavioral and metabolic functions, because they are the precursors of many of major neurotransmitters and neurohormones, and they are essential for the synthesis of ATP. And deficiencies in vitamin B3 have been associated to several disorders in humans, in domestic animals, and in livestock, such as pellagra in humans, or growth retards and aggressiveness in rodents, and also an impaired ability to orient in space in the honeybee. But what about wildlife? Well, the information is really scarce about wildlife, but we do know that plant content in tryptophan and vitamin B3 vary greatly, with plants that are really deficient in these micronutrients, such as corn and dandelion. And on the extreme end of the gradient, you have red maple, which is really rich. And in between, you have different species with flowers and wildflowers, and crops that have low to moderate amounts of tryptophan and vitamin B3, and trees and bushes that have higher amounts in their tissues. You can see that there is still a great variation in between bushes and trees, and I want to highlight important trees that we have here in Eastern Canada that have very high content. Regarding animals, we have few information, but some studies reveal that the consumption of corn or dandelion led to abnormal behavior and reproductive failure, such as maternal infanticide in European hamsters, or autophagy in European bumblebee queen, terrestrial bumblebee. Consumption of abundant or at least interesting levels of tryptophan and vitamin B3 associated with sunflower, increased maternal behavior and reproductive success in European hamsters, reduced aggressiveness in bumblebees. But we also know that sunflower pollen has medicinal value for bumblebees. And at the extreme end of the gradient, we know that consumption of red maple pollen improves the reproductive success and triggers reproduction of chipmunks. So now, how can we apply this knowledge to bee conservation in Canada? Well, to do so, we assess the nutritional and medicinal value of native plants pollen for bumblebees based on their content in tryptophan and vitamin B3. And unfortunately, we do not have results to share on the medicinal value yet, so we will focus mostly on the nutritional aspect only. The main limitation we do such kind of studies is to find the pollen, especially of wild plants, native plants. But fortunately, we found some collaborators, the two beekeepers that are also pollinologists and that could provide honeybee collected pollen from southern Quebec. So we managed to get 3 monofloral pollen of trees that have high content in vitamin B3, and we compare them with the commercial mix. And given the gradient, we expected that red maple will promote reproduction of bees.
Now to go into our methods. For our experiments, we caught 20 wild Bombus impatiens and 20 wild Bombus griseocollis queens from Southern Ontario. As you can see, mainly around the Greater Toronto area, and queens were collected at 19 sites in total. These queens were then transferred to our Bumblebee conservation lab at African Lion Safari. Queens were installed in BioBest queen starter boxes and were fed pollen every other day and nectar every 4 days. The first experiment we conducted was to see how red maple impacted the reproduction of wild-caught queens. Half of each species were fed with mix, which, as Mathilde mentioned, was a commercially available honeybee-collected pollen. Another half were fed with red maple. And the red maple pollen balls were 75% red maple and 25% mix. And then we looked at the proportion of queens producing workers, males, and gynes. And then we looked at the number of workers as a proxy for colony size. The second experiment, we looked at how Sumac and Hawthorne impacted the reproduction of Bombus impatiens micro-colonies. These workers were taken from the first experiment, and we made 45 micro-colonies, and each micro-colony had 5 workers each. Bees were fed 2 balls of pollen, 2 of mix in the control group, and 1 of each in the Hawthorne and Sumac treatments. So they actually had a choice between which pollen they ate. We investigated the proportion of microcolonies producing males and number of males and number of eggs produced. So we just have some preliminary results. This just finished a few weeks ago, so haven't had the time to dive in, but here's the proportion of colonies that produced workers. Red maple fed queens are in red, and the mixed fed queens are in orange. As you can see, impatiens and griseocollis queens fed red maple pollen were more likely to produce workers than mixed fed queens. This graph shows the proportion of colonies that produced males. Again, red maple colonies were more likely to produce males than mixed colonies. And this is shown with the proportion of colonies that produce gynes again. Again, red maple colonies were more likely to produce gynes than mixed colonies. And also you can note here that there's no impatiens colonies that were fed mix that actually produced gynes. Here shows the number of workers produced throughout the whole season for Bombus impatiens. Red maple had a higher average number of workers, 141, compared to mix with 76. And this is mainly due to the higher initiation success and red maple colonies. However, when you look at colonies that initiated, aka you take out all the zeros here, you can see that the few mixed colonies that did initiate actually had higher number of workers on average, 254 compared to 177 with red maple. This may be due to several reasons. It's pretty unclear whether it's actually solely because they were fed mixed pollen. But some other reasons might be is that they put all their resources into actually producing workers, since none of these colonies produced gynes. And also the majority of queens fed mixed pollen just happened to be installed a little bit before the red maple queens. So maybe if they were installed right after emergence, they might have higher reproductive success. Again, here shows the number of workers produced throughout the whole season for Bombus griseocollis colonies. As you can see, the mix actually had higher average number of workers, 8 compared to 6 for red maple, so this is slightly different than impatiens. This may be due to having a slightly different, slightly smaller difference in initiation success between the treatments. And then this is reflected in the averages for colonies that initiated. As you can see, the mixed colonies had a higher number compared to red maple. And again, mixed colonies having larger colonies may be due to several reasons, likely similar reasons to the impatiens colonies. Moving on to the results for our second experiment, looking at impacts of Hawthorne and Sumac on impatiens microcolonies. Here are the average number of males produced per microcolony. As you can see, there is no clear difference between the treatments, that's for sure. And similarly, there weren't any clear differences between the average number of brood cells per microcolony. Again, there are multiple reasons why we might not have seen any differences between these treatments. The main possible reason is that they were actually given a choice whether they wanted to eat mix or sumac or hawthorn. So maybe they just ate more mix than the other pollens. Another is that preferences still haven't been looked into, and other parameters such as pollen intake still need to be explored, since they may have had to eat more depending on the diet.
So to wrap up briefly, these preliminary results show a clear benefit of red maple for queen reproduction, but then we have to investigate more about the other results. We will now continue also to investigate pollen nutritional values with other pollen and bees to be tested next year. And we will also assess the medicinal value of the pollen that we used by screening all the bees and the workers and the larvae for viruses by genomics. And finally, one of the next steps is to evaluate the biochemical composition of each pollen. The ultimate goal is to find the complementarity between the pollen of crops and the pollen of wild plants to find associations that could be promoted in the fields or in the landscape, and that could offer all the benefits for bees. All this information will be directly transferred to bee conservation as part of a broader project. So this project involves a lot of people that as you can see on the slide. And the results that we presented with Sarah today will help inform concrete conservation actions that will be implemented in collaboration with farmers and with beekeepers. And this knowledge will be transferred to all other stakeholders and used to inform the public as well.
So yeah, we want to acknowledge everyone that helped us with this project from the Wildlife Preservation Canada side. I'd like to thank Ellen Richard and Tiffany Harrison for their great help catching all of our wild-caught queens and all of the lab work that they put in. I also want to thank our collaborators and funders.
And I'd like to thank our two collaborators, Yann Loranger and Isabelle Rabat, for providing the precious pollen, and the founders as well as my institutions. And thank you all for your attention.
Thank you so much. Excellent talk. Please do raise your hand if you have a question for the Mathilde Sarah combo. I see Clement has one. He kind of wrote it in shouty font, but I don't think he's shouting his question. Are there mid to late summer pollens that are high in B3?
That are high in B3, that's the question?
Yes. Yes.
Okay. Well, I don't know, because there are very few information about the content of the pollen. So I'm currently investigating that. So we are implementing a way to measure all the vitamin B3 forms in the pollen, and we will target different plant species. I guess that there are some, but I'm more aware of like the poppies, which is more a European species. So we'll have to have a look on the Canadian ones.